officially to our very first STEM Talks of 2025. So happy new year. Hopefully you all had a good holiday break. But we will be focusing on stem cells today. So if you have never been to one of our events before, essentially how our career panels work is we pick three amazing individuals that work in a specific field. And the topic, as I said before, is stem cells this month. And they will talk about what they do in this field. So what they do on a day-to-day basis, how they got to where they are, and then after they all present, we'll have an open Q&A period where you can ask your questions and they will answer as many as they can. As you can see, I am recording the event, so you can watch this back later, but because of that, just try to keep your microphones and cameras off to not interfere with the recording. For those of you, though, that are here just for your enjoyment, great. But if you're here for your enjoyment as well as the certificate program, in order to get credit for a certificate program, all you have to do is make sure your first and last name is spelled properly on Zoom. We will then take attendance from there. Additionally, we will have an exit poll at the very end of the event. It'll just kind of be a mini trivia. We will test your knowledge on stem cells. So make sure you fill that out at the very end. That'll happen right before the end of the event. For those of you that are coming to your very first event and have no idea what the certificate program is, I'll give you a little bit of an intro. In the meantime, Trisha will just launch a bit of a welcome poll so we know who's in the event today, if you do have interest in stem cells, if you do want to learn more. But for those of you that don't know what the certificate program is, essentially it's a program that our team, UHN STEM Pathways, runs, where students have the opportunity to achieve certificates for coming to our events. So if you come to 7 of our events from now until June, we'll give you a certificate of participation signed by senior UHN leadership. Additionally, if you are in the health and wellness program, the chosen program, we can also give you a certificate for medical terminology or lab practices. So all you have to do is attend 7 of our events, whether they be career panels, interactive activities, in-person events like PA days. There's a lot happening from now until June, so there's plenty of time to hit 7. So these just come out some of the upcoming ones: engineering, medicine, nursing, etc. All right, so if you have all filled out the poll, great. Then we're going to start the event. So I think, Melissa, you're up first. So whenever you are ready, feel free to take over screen share and talk about your work in the lab.
So hi, everyone. My name is Melissa. I'm a master's student in the FLAM Lab, which is part of the McEwen Stem Cell Institute at University Health Network. And we're also part of the Department of Laboratory Medicine and Pathophysiology at the University of Toronto. So I'm from Aurora, which is, if you don't know, a small town outside of Toronto. And I have been working in the FLAM Lab for just 4 months now. I started in September as a new master's student, but I've been living in Toronto for over 3 years as I started my undergrad at U of T as well. And in my spare time, something I enjoy is triathlon and singing. And also being a part of events like the ones at STEM Pathways. As you can see here, this is like one of our lab tours that we did in our lab. So as I mentioned, I did my BSc at the University of Toronto in human genetics and physiology, and I just graduated this past June. And then I'm now just in the thick of my master's at the University of Toronto. So I'm just going to talk to you a little bit about what my lab does, and as well what sort of a typical work week looks for me and what my project is focused on. So my lab in general, we're all about developing cell therapies for heart failure. So a lot of patients with heart failure, really the only option for them is as their heart muscle is failing is a full heart replacement with a transplant surgery. But as you may or may not know, there are limited supply of organs. And so one of the potential alternatives our field is focused on is basically taking stem cells and turning them into heart cells in a dish that we can then give to these patients to help regenerate their own heart. And so how we do this is we use genetic engineering techniques to modify the cells so that they can survive better, or they can be safer for patients, or they can function more efficiently. And so my project is exactly that. So one of the biggest issues is in models of this therapy, when we transplant these cells, they cause arrhythmias. And if you don't know what that is, that's basically when different areas of your heart muscle are beating out of sync with each other. And that prevents that good circulation of blood that you need throughout your body. And so one of the ways that we're approaching trying to solve this problem is we're engineering the cells to have an electrical safety switch. So something that we've given the patient this transplant, hopefully it's benefiting them, but then we start to see these arrhythmias and we can shut the cells off to stop them from hurting the patient. And so one of the ways that we do this is we use a lot of gene engineering techniques. So these are techniques to get into the cells and change their genetic code. So we can change the different proteins that they're expressing, and that ultimately changes how the cell works. And so one of these techniques you might or might not have heard of is CRISPR. And so this figure just kind of shows our workflow. So we have these special proteins that are acting as the safety switch. We introduce them to the cells, and then we turn those cells into heart muscle cells. And then we transplant them in these models to see if they're working like we expect them to. And so what does all that culminate in? Like, what's a typical week like for a master's student? A lot of it is experiments and cell culture. So I make cardiomyocytes, which are those muscle stem cells. I do a lot of cloning and gene editing techniques, and then some other experiments to take a look at my cells and make sure that they're functioning the way that I think they should be. And then I also spend a lot of time self-learning. As you know, as a graduate student, a lot of the learning's on you, right? You have to go out and read papers on your own and figure out what you need to know for each experiment. And then there's a lot of data analysis involved. We have lab meetings and presentations as sort of a way to check in and make sure you're on track. And then, of course, there's the course side of being a master's student. So you spend time at the university attending lectures, learning about other people's research. And there's a whole writing side of applications. And then there's also a lot of ways when you're a graduate student to kind of get involved outside of your degree. So some of the ways that I do that is I volunteer at a hospital nearby. I spend time doing triathlon. I love, reading and music, and I spend time with family and friends as well on the weekend to kind of have that balance. So if you have any questions about like sort of the lifestyle of a graduate student, I'm happy to answer those as well. And so just to kind of close it off, like what's the importance of what I'm doing and why am I on this path? Really, I mean, one of the things that excites me most specifically about my project is that these arrhythmias are like kind of a major barrier to cardiac stem cell therapies. And so we're hoping that this might be a small piece of the puzzle to kind of try and help address that. And sciences in general is a challenging discipline, but it's also incredibly rewarding. You get to be curious, you get to explore, and you get to contribute to like kind of our general understanding of how biology works in the world. And in terms of my general plan, I love research. It's something I'd like to do throughout my career. And then I'm also working on pursuing my medical degree because I'd love to be in contact with patients. So yeah, I can also answer questions about sort of like balancing those aspects. I'll pass it off to the next presenter.
Thanks, Melissa. Alex, you are up next, so take over screen share.
Hopefully you can all hear me. Yes. So hi, my name is Alex. I'm a PhD student here at the UHN Stem Cell Institute. Specifically, I'm a part of Christina Nostro's lab, and I'm also affiliated with the Department of Physiology at the Faculty of Medicine here at the University of Toronto in the downtown core. Yep. So I was born and raised in the GTA, and I've been working in the Nostro lab specifically for about 3 years now. Previously, I did my undergraduate studies at the University of Toronto as well. And during my time, during my undergraduate studies, I also did research in labs located at SickKids and also Mount Sinai here in downtown Toronto. On my off time, I also enjoy traveling and also reading fantasy novels. And here you kind of see the two aspects of my life right now. Either I'm in the lab doing cell culture or I'm trying to do fun, exciting things like traveling. So I got my honors Bachelor of Science at U of T. I specifically specialized in human physiology with a collaborative major in health and disease. And I always knew I loved biology since high school, really. But my undergraduate studies really helped me focus in on my specific passions for regenerative medicine and stem cell biology. So now I'm pursuing my PhD in those exact fields. So after my undergraduate studies, I decided to do a direct entry PhD program. So what do we do? So the Nostro lab in general is really interested in directing stem cells to become cells of the pancreas for the purposes of diabetes research. So as you heard from Melissa, stem cells are really awesome cells that have the potential to become anything that we theoretically want them to be, as long as we give them the right instructions. So while Melissa was turning them into cells for the heart, we're turning them into cells of the pancreas. And this would allow us to study the development of the pancreas, explore potential cell-based therapeutics for diabetes, or even investigate disease modeling. So to this regard, I'm going to talk about my specific project. So you may have heard of other forms of diabetes, type 1, type 2, maybe even gestational diabetes, but I'm specifically interested in cystic fibrosis related diabetes. So cystic fibrosis, you may or may not have heard of, is a multi-organ genetic disease. And thankfully, people are living longer than ever before. But as people are now living longer and having to live longer with this disease, there's a greater incidence of secondary complications that are occurring that need to be better understood. And this form of diabetes is one of the most prevalent complications that's troubling the CF population as of now. So in an attempt to better understand this disease and explore what's going on, I'm specifically using stem cells that are actually derived from cystic fibrosis patients. So we can take cells from CF patients, reprogram them to become stem cells, and then we can use them in our context to look at development or disease progression in the context of cystic fibrosis and our differentiations. In addition to this, as I mentioned, we're interested in turning cells into different cells of the pancreas. So I'm specifically interested in also looking at how to turn stem cells into the duct portion of the pancreas. So these are some pictures of the cells I've been generating. So this is a new novel thing that I'm working on. So all in all, the work that I do encompasses disease modeling, looking at patient-specific contexts, and also looking at new ways to generate new cells. So a week in the life for me, it includes a lot of things. It can vary from week to week depending on what's going on, but generally there's always experiments going on that include cell culture, so making the cells, analyzing the cells. There's a lot of planning that goes into doing your experiments before you even conduct them. So there's a lot of organization that goes into it. A lot of literature review, so that includes the self-learning aspect, whether I'm reading papers, discussing with other people about what's new in the field. Lab meetings, so we often come together as a lab to discuss our progress, new things that we should be on top of, and stuff like that. There's data analysis, so the data that we generate with our experiments, we have to analyze them to really understand what's going on. I have one-on-one meetings with my supervisor, so this really helps me organize myself, see where I should be going, what I can improve on. There are a lot of seminars that go on that we can attend, whether that's from the University of Toronto, other institutions, hospitals, etc. And on the kind of extracurricular aspect, I'm a part of the Graduate Association for Students in Physiology. So that includes extracurriculars, association meetings, event planning, and attending those graduate socials that I do organize. So whether that's a pub night, skating at Nathaniel Square, or a chill paint night at one of our institute buildings. So importance and why I chose this career. I would say it's very obvious how research and investigating science can be very exciting. We're actively participating in improving the quality of life for patients. However, there's also a big aspect of that it brings awareness and builds supportive and collaborative communities, which I'm very thankful for and excited to be a part of, whether that be within our scientific community or the greater healthcare community. And I chose this career path because I'm passionate about actively contributing to those landscapes, but also it offers me an opportunity to be innovative, creative, and constantly learning in an environment that's both very highly independent, but also very multidisciplinary, as in that you will be contributing to a lot of teamwork and aspects. And further down the road, degrees in such fields like biomedical research, which is what we do, offers you a wealth of opportunities, both locally and internationally, in academic, industry, and also government sectors. So there's a lot of opportunities. Thank you.
Thanks, Alex. Adriana, you are up last.
Awesome. So, hello everybody. My name is Adriana Migliorini, and I am a scientific associate in the Nostro laboratory at the UHN STEM Cell Institute at UHN. So, I am from Florence, in Italy, and I lived in Germany for 7 years. And at the moment, it's already 7 years that I live in Toronto and work in the Nostro Lab for around 7 years and a half. And in my free time, I enjoy traveling, hiking, and Pilates. I did my bachelor and master degree in medical biotechnology at the University of Florence in Italy. And for my PhD, I traveled to Germany and I did my PhD in human biology at the Ludwig Maximilian University in Munich. So the topic of my work is pancreatic development, so similar to what Alex is working and told you about before. And to do so, I use human pluripotent stem cells to model development in the dish. And in particular, I study the role of immune cells during pancreatic development. So the big question that my research is aiming to answer is, how can we create insulin-producing cells that work just like the ones in your body? And how can we protect these cells so they can stay healthy and keep producing insulin when we transplant those cells? And in particular, I study a special kind of immune cells, which are called macrophages. And macrophages are sort of like constructor workers that can help insulin-producing cells to grow and work better in certain conditions. And what my research have found so far is that macrophages, they can send signals to help insulin-producing cells to develop and survive. And as well, these signals might help the vascularization of the insulin-producing cells when they are transplanted. But why all of this matters? This is because this research could actually make cells therapy a reality for people with diabetes and improving their life and reducing the need for daily insulin injection. And if we take a look at my life during the week, it's quite busy. So I do a lot of lab work, mostly conduct experiments and to culture and differentiate those human pluripotent stem cells, and as well co-culture them with macrophages. I spend a lot of time analyzing the experiment that I perform. Also, a good part of collaborate with colleagues to design new experiment, refine protocol, and brainstorming. As well, I spent some time on mentorship, so guiding junior colleagues or interns and sharing expertise on stem cell culture and 3D system and macrophage solid interaction. As well, I write and edit a lot of sections and a grant proposal for manuscript, and as well, taking part of meetings and giving seminars. And of course, I also have some personal time, and I'm trying to balance, some professional commitment with time with family, for family and friends. And if you ask me why I chose this career and the importance of this career, is mostly because I love the idea to develop innovative cell therapy to improve diabetes treatment, and as well as advance this understanding of islet development and macrophages, how they can support this. And also the importance of bridging biology and medicine to address critical health challenges like type 1 diabetes. And of course, also inspiring the future scientists through outtrack and mentorship. And why I chose this career is because I'm very passionate about stem cell biology and regeneration. And I love this interdisciplinary problem solving and innovation. And as well, I'm committed to advanced science and mentoring others.
All right. Thank you everyone for talking about yourself. So now is the open Q&A. I'm going to bring everybody back onto the screen. In the meantime, hopefully everyone has seen we have a new Q&A button set up here on the webinar. So if you do have questions, feel free to just submit it to the Q&A part of the webinar, and we'll try to answer as many as we can. As you guys are adding questions, we're going to start off with some pre-submitted questions. So students have been submitting them as they've registered. And the one question that I saw a lot was a nice little general one to start with. Why even stem cells? There are so many fields in STEM. Why did you pick stem cells? Why did you want to do a graduate degree and even further in this field? And maybe just for simplicity, we'll go in the order of the speakers. So Melissa, why stem cells?
Absolutely. So I did my undergrad in genetics, as I mentioned. And actually a big part of my degree was learning about stem cells and what makes them so special. Why can they turn into all these different cell types, and what are some of the ways that we can use that to make new discoveries and to help people? And I just found it really fascinating. I also, I really enjoyed when I was learning about my lab, how they use these genetic engineering techniques to try and make stem cells better for therapy. That was something that I really wanted to focus on and be able to learn. So yeah, that was what I really enjoyed. It's a very exciting field to be in because it is very much sort of, I think, at the forefront. A lot of this is very experimental, but we're trying to progress it closer and closer towards the clinic, which is really cool.
Alex, what about you?
Yeah, I guess it started in undergrad, when I was being taught about human development, and that kind of pivoted me towards the idea of stem cells and what they are capable of and what they do. So stem cells piqued my interest because they kind of take you all the way from human development to also kind of the end stage. It gives you a nice snapshot of studying how certain cells and certain organ types form in the body, towards also how you can use those cells then to treat those organs and bodies. So it kind of bridges everything that I was interested in. It's a very interesting and relevant topic right now. If you think about things that have direct or soon to be direct clinical implications, we already have stem cell based therapies that are being tested in clinical trials. So there's a lot of room for interesting and innovative work to go on.
Adriana?
Oh, yeah. Well, I think Melissa and Alex cover most of the answer why stem cells, and yeah, I mean, I can just add from myself. I'm a big fan of human development, and to understand how organism and cells develops, and stem cells, they really offer this opportunity to model this process in vitro, to understand the mechanism, and then further on to manipulate those those pathway, and as well also to maybe also improve it, right? So to generate cells not in 9 months, but maybe in a week or a month. And this is where our research is mostly focused. And of course, then for their clinical application, which is just, I mean, amazing. So yeah, that's it.
All right, we have like a general question. Can someone just very kind of simply explain what stem cells are? I guess a lot of the students heard what you were saying, but they want maybe a more simple version of what stem cells are, and how exactly you use them. So who do you think might be able to simplify the most?
I can do that.
Take it away.
Okay. So stem cells are cells that basically can become every kind of cells in your body, right? So if they're instructed with proper signals, and this is what we do in the lab, so we tell them which road to take, then those cells, they're able to generate each cells of your body, like epithelial cells or cardiac cells or pancreas cells. So that's the easiest explanation I could ever give. So they're very plastic. They can be everything. They can become everything when properly instructed.
I guess kind of connected to that, they have noticed that some of you are working on creating heart cells versus different types of cells. So how did you pick which branch of stem cell research were you interested in? Did you just pick any lab that would take you, or did you pick one specifically based off of that branch of stem cell work? And maybe Melissa, you can go first.
Sure. No, I did kind of pick specifically. I was really interested in the heart and it's just its physiology, like the way it works. I think it's a really cool organ because it has like these electrical components, right? You need these electrical signals for your heart to contract. There's also all these like hydrodynamics components. You have to have the right balance of fluids and in your chambers. And so I think it's just a really cool system to work on. There's a lot of different things going on at once to make everything work together. And my project is really focused on electrophysiology, which is how those electrical signals turn into like the cells contracting and your heart beating, essentially. And that's something that I found really, really exciting.
Alex?
Yeah, that's a great question. There are a lot of labs just in Toronto that do different things. During my undergrad, I did my work in a lab that works on lung cells, so turning stem cells into lung cells. So I made my way over to the pancreas largely because U of T actually has a very rich history with stem cells and also diabetes research. So as an undergraduate, I was exposed to a lot of content and coursework related to endocrine pathways, metabolism, diabetes, insulin, etc. So whether it was accidental or not, I kind of made my way to finding a really deep interest in endocrine cells and the pancreas. So I purposely made my way to the current lab that I'm in because it kind of merged both of those things that I was looking at, endocrine and also stem cell research.
And Adriana.
Yeah, so I actually started off with my PhD studying kidney stem cells and so on. And then I moved to the pancreas because I wanted to explore different organs and learn more. And also because this was actually available at that time in the city, and there was this amazing lab starting off with this research. And so that's why I said, yeah, why not?
I mean, stem cells are cool in all the branches, so.
Yeah.
All right, we have a lot of questions, I guess, about your skills and experiences, so I'll try to kind of group them. So I guess the first question was, if people are interested in research, are there any things that they should, work on, any skills they should develop? Like, how should they start thinking about research and getting into research in the first place? Melissa, you can go first.
Sure, absolutely. I think, one of the biggest and easiest places to start, I think, is just YouTube. I mean, go online and see if you can find like scientists being interviewed. There are some great like science channels that explain research in simple terms. And that's a great place to start because it helps you kind of understand like how science works and how we try and interpret the world, and some of the language that we use as scientists. And then if you're a step above that, if you're like, I've already got that covered, I'm curious, I have kind of an idea of what I'm interested in, you might want to try, there are some research papers, a lot of them that are open access. You don't have to be a part of a university to download it. You might want to try going to some research papers and just download one and try and read it. It is going to sound like gibberish the first time. My first research paper, I was so confused. I was like, how am I ever going to do this? It gets so much easier. Science is just like another language. It's like learning French or something, but it gets easier a lot faster. I think the learning curve is a lot quicker. And that's something you can maybe also like bring to your high school science teacher. Be like, hey, I found this paper, and I think it's really interesting. Can you help me understand it? And they might be able to help you kind of walk through it. So I think those are some of like the easiest ways to start, just like dipping your toe in and learning how to understand science.
I thought that was a good answer. Alex, how about you answer the next question I have? The
Sure.
question is kind of related. It's more just on the extracurricular side of things. So like how would you, what should you join? Should you join any clubs? What should you do on that side of things instead of Googling or researching papers?
Ah, I would get like I guess the question is more like in a high school context. Um, Honestly, I would say for someone starting out, if you are interested in research, specifically, you don't have to worry too much, I would say, about gaining science specific or research specific extracurriculars or experiences under your belt. When people are starting off, they're all kind of in the same boat of like they don't have the research experience because they don't have access to high-tech facilities and laboratories. I would focus on building like your core and interpersonal skills. So if you can show that you work well in a team, that you are an organized person, that you are reliable, etc., whether that's being a part of sports teams, extracurricular clubs, band, orchestra, etc., it shows a level of discipline and commitment that's very important and translatable in research. I knew a professor, for instance, during my undergrad, who was talking to us about this kind of content, and she was telling us how she was really impressed by one particular applicant because she was, I think, in youth orchestra for like 10 or 8 years. So that really showed the professor that she's committed, she's passionate about things that she's interested in, So these are all translatable things that can be applied to not just research, but anything else that you're interested in. So you can really sell what you're already doing. You don't have to worry about necessarily having specific experiences in whatever you're interested in at the moment.
It's like the perfect answer. Adriana, I have a question for you specifically. They are asking, what was it like studying and working away from home for your schooling?
That's an excellent question. It was amazing. It was a great experience, especially for my PhD. Was really nice. It was fantastic to have the chance to experience a new culture, new environment, a completely different languages, and in particular being in the lab, it was sort of magic because in the so our life as a scientist, we really spend so much time in the lab that your coworkers and your friend, they become your friends and part of your family. So, and when you're abroad and when you're outside, far away from home and so on, then they really become a sort of a community and it was great. And especially be exposed to this different culture and different environment and or also scientific environment. The way they do research in Germany is very different compared to what they do in Italy or what they do in Canada. So I think I was very fortunate to have the chance to experience all this different environments. And it was very enriching, especially from a scientific point of view and for knowledge. So I would recommend to everybody if you have the chance.
All right. Well, in terms of studying locally, we have questions for our U of T experts, I guess. What is undergrad at U of T like, or grad school at U of T? Is there a lot of lab work? Is there a lot of courses? Is it hard? They just want to know, should they go to U of T, basically? So, Melissa, you can go first. Tell us.
Sure, absolutely. I think I can definitely speak to this because I was an undergrad and now a master's at U of T. I think U of T as an undergrad, if you are interested in research, it's a really great place to be because there's so many research hospitals. There's just so many opportunities here. Um, and that being said, U of T is a big university. So you do have to be aware of like what works best for you and what kind of environment you want to be in, right? I think with U of T, it is challenging academically, but it's not impossible. Don't let people on Reddit or whatever dissuade you. You can absolutely do it. I think in university, it's all about just realizing that if you want help, if you want resources, if you want to make friends, you have to reach out. That's the biggest thing because the resources are all there for you. It's just no one's gonna like in high school like tell them you and hand it to you. So there's all kinds of like academic supports at U of T. So if that was a concern, I would say don't be dissuaded. And then the other thing is, I think life at U of T, it's really amazing because there's so many opportunities. You're right in the middle of the city. And so it's just about trying to find those little communities where you fit in and you feel like, okay, you've got something you're passionate about, right? So it's about building sort of community in this big city. So I think there are trade-offs, but ultimately I loved living in Toronto. I loved being at U of T and participating in like sports and academics and music and all of that. And having all those facilities there for us. And then also I did a lot of research in my undergrad. It really helped me to get into grad school. And I really enjoyed my time at UHN as well. And then in terms of grad school, I would say, especially for stem cells, it is long hours in the lab. It is a lot of hard work. And at least for LNP, it's relatively few courses in comparison. So most of your time is going to be spent in the lab working on your project. But it is very rewarding work if it's something that you love and enjoy. So I would highly recommend it. And again, it's pretty much the same as undergrad. Like being in the city, there's so many wonderful opportunities and resources there.
Alex, anything to add?
Not really. I'd say Melissa covered it pretty well. Um, yeah, I know U of T has a reputation, I would say, depending on if you ask like your high school teachers or whoever. Yeah, it's not easy. I'm not going to say it's easy, but yes, it's doable. And there are ways to make it easier for yourself, like Melissa said, by building those communities. U of T is ginormous, but it does, but for better or for worse, kind of force you to kind of build those communities, whether that be through tutorials or smaller classes or U of T Arts and Sciences specifically does a thing where you are split up into colleges. So that's like a smaller funnel, finding people in your classes, things you do together, lots of clubs. There's a lot of stuff going on at U of T and at such a very high caliber that you'll be able to find what you're interested in a very small, relatively small environment.
All right, we have questions, I guess, more about kind of the lab work and lab work environment. So maybe Adriana can go first. The first question is, you're working with these very fancy cells. Are you ever nervous that you're going to mess something up or do something wrong? What will happen? They want to know.
Of course, every time. And we mess up also very often. No, I'm joking. I mean, making mistakes is normal, and it can happen, right? And we have security rules in place and so on. So this is not I mean, it's not a huge deal. What is very important, and I'm always telling to everybody and also to myself, is that when you make a mistake, it's not the end of the world if you can recognize it and you can learn from it. And actually, this is the best way to learn. So, especially, during your PhD or your undergrad or your first research experience, if you have the chance to make some mistake on the way, actually, this is the best way you can really learn because you learn to troubleshoot. So yeah, would be my answer.
So you will be nervous, but it's okay.
Yeah, it's okay. It's normal. We are researching, right? So that's part of the job.
All right, next question is, what is kind of the best part about working in a lab and also the worst part of working in a lab? They want to hear both sides of the spectrum. So, Melissa, I keep picking you to go first, but go again.
Okay. I would say I think like the best part I'll start with is if you're someone who's really, really curious and really passionate about like just how biology works or how the world works, I think it's a great place to be. And also if you're someone who's very like who just enjoys like kind of that self-driven learning, I think it's a great place because you have some kind of authority over what you're doing, and you get to be a part of this sort of creative design as well as the sort of science side of it. So that I really enjoy. In terms of the worst part, I guess I don't know if it's a bad thing. I think it's just be prepared that it is likely going to be long hours. It does vary somewhat within like different disciplines, but it is a lot of work. It is a lot of work to do grad school. And then the only other thing, this isn't a downside. I think it's just an important thing to keep in mind down the road when you're looking at labs. But even as an undergrad, when you're looking to like participate in research, one of the most important things I would say is the lab culture, right? You're going to be working with the same small group of people every day for years, right? So it's important that they're people who, can mesh with your working style and who you can get along with, and who have a positive way of teaching each other and dealing with mistakes and things like that. So I think, but if you can find like a good group of people and a good lab, it can be a really, really fun and wonderful experience. You also get so close with your coworkers, which is a wonderful thing as well.
You become your best friends. Alex, what about you? Best and worst?
I'd say the best thing is that um, I'm always doing something exciting. So sometimes your everyday it can be very easy to think about it as like an everyday thing. But when you sit back and really think about it, you're doing something really exciting right in front of you. I'm turning one cell into another cell. There's a lot of exciting science going on. So that's my best part. I guess the worst part is that when you're so hyper focused on something, sometimes your perspective on things can kind of pigeonhole you into a certain aspect of your life. So it's always good to take a step back. It's very easy to find yourself not like spiraling down into something, but maybe too hyper focused on one thing that you might forget about something else that's also important. So assessing what's in front of you is very important, and having those people around you that help kind of maybe pull you out of the downward spiral is really great. So your lab is also very important. But yeah, that's I guess the best and worst aspects of what I do.
All right, last panelist, what about you?
So I think the best part for me is that this job is extremely creative, right? You can be creative. You can think, you have to be really creative. You have to We try to think to solve a problem and think out of the box and so on. Another great thing is that every day is always different. It's never the same. It's literally never the same. You plan something and then things go completely different. You look at the date, oh my God, I have to do this, this, this. So it's really nice. It's very creative and it's also very dynamic, right? I mean, I won't think scientists is one person job, but it's also a community, right? So you have your lab members, you talk, you discuss things and so on. So this is great. The worst part of our job is that discovery takes forever. It takes very long. So you really need to train yourself to not be discouraged. To not be discouraged if you have you have an idea, you have a question, you have an hypothesis, and then you do the experiment, and then turns out completely different, and so on. And first of all, it turned out in a way that could answer, so which is already great. And then you just to pick up from them. So you really need to be patient. Yeah, discovery takes years. It's not something rewarding like there are many other jobs where you have a task, you get the task done, and then it's done. Check. And research takes years over years. So yeah. So, but that's also the beauty about it. Yeah.
All right, then speaking of research, we have a lot of technical questions now about stem cells. So if everyone's ready to use their expertise, we'll start off with the simple one of how do scientists term stem cells into the specific type that they want to work with? So anybody want to start with the general? How do we make a specific type of cell from stem cells?
I can start off and then I'll hope like you guys will add to whatever I've missed. But I think one of the ways I like to sort of think about it is I mean, I know we have maybe varying degrees of knowledge about cell biology, but basically there are different types of cells in your body that have different roles, right? Everyone has a job. Like the heart cells have to beat to make your heart work. You have neurons in your brain that have to fire and help you to think and coordinate your movements, and muscle cells that have to respond in your arms and everything. And so when you have a stem cell, it can become any one of those, right? It's nothing yet, but there are certain little like signals, little cues that we can give these cells that say, oh, hey, you're supposed to do this. You're supposed to start, wearing these certain proteins so that people can know you're this type of cell, or you're supposed to start doing this job so that you can start acting more like this type of cell. And there's different stages throughout that process where they get more and more like that type of cell. And so a lot of it comes down to when we're doing this in a dish, a lot of it are just these little like signals that we feed them. But then there are some other things that they also get in their environment that they maybe don't get in a dish, and we're trying to incorporate that, right? Like you might imagine that in the heart, like cells start beating around them, these cells start getting stretched, right? That's a mechanical little signal to tell them, oh, hey, I'm in the heart. I better start acting like a heart cell. And there might be electrical signals, right? They might be getting signals from their neighbors. And so those are, I think, in some of the newer like ways that we're trying to turn cells into more and more like the stem cell or the cell, sorry, that we see in the body. We're trying to incorporate all these other different like cues that we can use.
I think that was a good overview. All right, one of the next questions is, where do we even get stem cells from?
I think I can answer that question, maybe. Or if Alex wanted to answer, go for it. Okay. So where we get stem cells? So there are mainly two major sources of stem cells. One are the embryonic stem cells, which are cell line, which have been derived many years ago, and those have been derived from the inner cell mass of a developing embryo at a certain stage. So this is the first source, and then the second source is we actually can generate stem cells, or also known as human pluripotent stem cells, by inducing particular factors into regular somatic cells. So if we overexpress certain factors, which are known also as Yanamaka factors, because he discovered them, we are able to reprogram those cells to get back on time and become pluripotent stem cells, and then be able to generate each cells of your body.
That was good. All right, Alex, you get the default, whatever the next question is. Hopefully you are ready for this. What are some cons of using stem cells? Are there cons of using stem cells? Good luck.
Cons of using stem cells. Um, I guess the biggest thing I could say is that they're very finicky. So because they have such huge potential, it also requires a high level of specificity and intricacy in what we do. So no matter how much we want them to turn into one cell type, for instance, there's a lot that goes into turning them into that singular cell type. And even following that process, it can go wrong. It can go a different way very easily. So it's up to us to kind of control that, even though it's sometimes really hard to control. So that's, I guess, one of the biggest cons. So there's a lot going into maintaining the cells, giving them what they need. And also because they can become anything, we have to narrow it really exponentially. And that narrowing, there's a large margin for error. We've done a great job so far in over the years narrowing that kind of perspective and really honing in on our expertise and our precision. But there's still need for work and improvement in that field. And I don't think that's ever going to change. We're going to be constantly improving that.
Great response, despite me throwing the question at you. All right, next question then is, so stem cells sound expensive. Where do you get the budget for stem cells or research? Where does money come from? How do you get money? Maybe Adriana, you are the most senior here. Where are you getting money?
That's an excellent question. And yes, stem cells are very expensive. So we get funding from either from the government, right? So we write grants and so on. There is a huge investment in Canada in stem cell research. And as well, we also get grants and funding from individuals. So from charity or from private foundations. And yeah, stem cells are very expensive, but I also have to say that there is a huge effort from us as a scientist also to make stem cell research and stem cell therapy less expensive, because if we manage to cut the cost, we're gonna make this cure or this therapy available to everybody. Otherwise, it's just gonna be a niche therapy, and this is something that we are also working on it. But yeah, most of the funding they're coming either from private individuals or charity or from the government as well.
All right, next question. I guess you talked about how we're making cells, certain types of cell types from stem cells. Some students are wondering, can you accidentally make cancerous cells in that process? Is that a risk of stem cell research that you can cause cancerous cells to be formed? Anybody want to answer that?
Yeah, I can go. Yeah, that is a risk. Once again, because the cells can become anything. If we're not precise enough, then anything can really come up, and that could be a cell that kind of goes crazy and then makes this like big tumor or etc. So in our research, there's a lot of work that goes into studying that and what can go wrong, so that before it even makes it into a clinic or to an actual person, we've done all our due diligence to make sure that that is minimized to as zero as possible. But yes, the possibility does happen. We see it in the lab when we do experiments. We see that kind of stuff go wrong. So that's why we do our research, really. Part of other people's research is to prevent that from happening, for instance. But yes, it's possible.
All right, next question, I guess still on like the worry safety aspect of things. A bunch of students are asking kind of ethical guidelines. Are there ethical guidelines using stem cells, preventing certain gene editing technology to be also incorporated in stem cells, all these things. Anybody want to tackle the ethics of stem cell work?
I can start off, but it's a very big question. So thanks for the question. It's huge. So before we start to talk about, like the risk and cancer and so on, I just want to point out one thing. So so if you take a stem cells, right, embryonic stem cells, human pluripotent stem cells and so on, you transplant this into an animal and so on. This doesn't really make a cancer like you might imagine, but makes what is called teratoma. Okay, so teratoma is actually a benign form in which you can actually have the formation of each of all the germ layer, so endoderm, mesoderm, and ectoderm, and also teeth and so on. So it's a little bit different from what you can imagine or what you think, like a cancer which stems from a somatic cell, so from an adult cell. So, and this is something that one has to take into consideration. I think we have a lot of rules and strict safety regulation in Canada and as well in the US. And when we have any cell product, any protocols, for instance, right? So we go through a very thorough screening, right? We use genetic tools to make sure that those cells, they are like all differentiating. And most importantly, we make sure that we do not, so that maybe we don't have 100% of the cells that we want to have, but we do not have any stem cell reside cells, right? So we don't have any of those cells that can become everything in terms of being controlled and so on. So we have a long way of safety first, and then we look for efficacy, and then as well then for therapeutic. So there are many, rules and regulation, and then we think twice, even maybe 100 times before to go into humans and so on. But so far, for instance, in the pancreatic field, the use of human pluripotent stem cells to generate insulin-producing cells has been shown to be safe and also quite efficacious. So individuals are transplanted at the moment with those cells, and they show independence. And now this is already ongoing since many years. So yeah, it's a long process, but we are trying to be as safe as possible.
Yeah, basically there are a lot of guidelines and procedures that have been approved by many boards and individuals, and then all the teams follow these to a T, so there's no concerns there. All right, we're kind of running out of time, so maybe we'll ask two wrap-up-y questions. So the first question is, so everyone's talking about, forming certain treating certain diseases, hearts, pancreas, etc. So can we use stem cells to make a whole organ or replace a whole limb or anything? Is that possible now? Is that something that could happen in the future? Can we recreate a whole organ? Anybody?
I guess I can start off and then I hope people can add. Yes, absolutely. I mean, no, is it there yet? I don't know. But in our field, at least, I know that there are many people that are trying to do that. They're trying to find ways to take stem cells and take like a scaffold and make an entire organ for replacement. I think at least in the heart, we're a long way off. And that's why our lab is focusing on some sort of smaller aspects, like can we just replace a little patch of muscle? But it is very, very exciting research that we hope will come to fruition one day.
And I guess kind of building on that, so what would be the steps to turning, your stem cell therapy into an actual clinical treatment? So like, where does your research go now? Does anybody want to tell me how their research gets into a clinic eventually?
I guess I can speak very generally in our specific context, for instance. So for those who don't know, diabetes is, I guess there's different types, but if we're thinking about type 1 diabetes, patients require insulin injections. So they're supplementing their health with insulin injections that are not produced by their body. So there's a very specific cell type that makes insulin in your body, and type 1 diabetics, these cells don't work. So that's why they need it externally. Our lab is working to generate those cells. So if we are able to generate those cells that make insulin, which we technically do, and we can put those cells into a type 1 diabetic and that produces insulin inside them for them, then they will no longer need to inject themselves with insulin. So that's kind of the idea and thought process that goes into taking a cell that we make and moving it into the clinic. There are efforts to doing that right now. There's still improvements that need to be made, but that's kind of one of the goals. Whether you're talking about cardiac cells, even retinal stem cells, et cetera, there's a bunch of different applications. But largely, you can think about cell therapy as in like we can make a cell and what need can it serve to replace or supplement in a context where it's not there normally.
All right, I think I'll ask one quick question to wrap up. In the meantime, Trisha, can you launch the exit poll? So as they're answering the last question, we have our exit poll, which as a reminder, you must complete to get credit for the certificate program. It's just going to test your knowledge to see if you have vaguely listened to what our speakers have talked about, like what is a cardiomyocyte, etc. So hopefully you do well, but please fill that out as we do our last question. And the last question is just give us a one sentence piece of advice for the students in the call. So I know, short, one sentence. Melissa, you're up first. Good luck.
Oh my gosh. One sentence. I guess just two things. Number one, just like stay curious, try to keep enjoying learning. And number two is just talk to people, do things like this. Experience being passed down is just one of the greatest ways to learn and jump over some of those big hurdles that you might find on your way.
Alex, your turn.
I'd say don't be afraid to try. Failure is a great way to learn. Being told no is a great way to learn how to improve. So never be afraid to try anything or try for anything.
And our last panelist, what is your advice?
So my advice is that science is amazing and just keep culturing, or nurture your enthusiasm for discovery and for asking questions.
The perfect little ending statement. So I'm just going to do one wrap-up slide, but thank you, panelists. This was actually, we covered so many questions. You answered all of them, so great job there. But I will do our wrap-up slide, so hopefully everyone can see my slide. But yes, that is the official end of our STEM Talk Stem Cells event. Hopefully you enjoyed yourself and actually learned something new. I think a lot of you are voting yes, so that is very good. But if you do want to kind of learn more about STEM, explore STEM in other ways, our team does run a lot of programming, both virtually as well as in person. And you can learn all about that on our website, UHN-STEMPathways.ca. And you can also just email us if you have any questions and we'll try to address that throughout email.